All right, so for our DNA lab, on your handout, there's some introductory material on the first page that just goes over what we've talked about DNA. On the second page is where you're going to cut out your paper model and get to play with pieces of DNA. So print out the other sheet that's in Canvas, that's the DNA components. You may need to print a couple of those out to have enough pieces. And notice that, so I've got some pieces here. So you'll have your pieces cut out. These, I already taped those together. So those would be separate pieces. And you're going to build the nucleotides. Remember, the nucleotides are the components or the monomers of the DNA. So the DNA nucleotides are going to be marked with a D. D is for deoxyribose. And then, so that's the sugar. And then the phosphate has the P. And then they're going to have their bases attached to them. So you want to build together your nucleotides. Just with tape will be fine. So this is one nucleotide. And then I have other nucleotides that I've put together up here. So now you're going to build your DNA molecule. And I've given you a sequence on the sheet to go by. So we're going to start with ATG. So here's an A base. So A, find a C, C, G. So ACG will be the first part. You can continue on into the rest of it. But notice that's only half of your DNA molecule. Remember, DNA is a double strand, so we've got to build the other strand that matches up to these. I didn't give you the other strand, but you should be able to figure it out because it's the complementary strand, which is going to match up. So what matches to A would be a T. And then notice on these pieces, well, notice how at first I didn't show you. Notice how the phosphate and the sugar fit together in a certain way. So make sure you're noticing that because these molecules have shape like puzzle pieces and they have to fit together in the right way. And then notice that as the A matches up with the T, it's forming hydrogen bonds. And A and T form 2 hydrogen bonds, so those are on here. But notice this is also going to make our sugar and phosphate on the other strand go the opposite direction. We mentioned this in class, how the two strands, one is the sense strand, which runs in the forward direction. The other is the nonsense strand that runs in the opposite direction. So that's why the sugar and the phosphate are going to go backwards on this side. But what matches up with a C here would be a G. So we get the G and match it. And notice it's going to form 3 hydrogen bonds. So that's going to match up. And then we need a C to match with the G. So that is also going to be the 3 hydrogen bonds. And you can continue on with the rest of the sequence. Pause the video and build the rest of your molecule. All right, now you're going to replicate, this is in part 2 on the handout, you're going to replicate your gene or your DNA. So remember, when the DNA polymerase enzyme comes in here, it's going to separate the 2 strands, and then it's going to match up nucleotides into this gap to build the copies of the DNA. And hopefully I have enough pieces. All right, so now we have 2 molecules of DNA, 2 copies. They're exact copies, identical copies. This is what happens during the S phase of interphase before mitosis, before the cell divides. It needs a copy of the DNA, 1 copy to go in each cell after the cell divides. All right, now we're going to move into what happens in transcription. So over on page 3 of your handout, we're going to do transcription with this DNA model. So you're going to need your RNA pieces. So I have my RNA pieces that are on that other sheet of the handout, or other sheet of the that you can print out. Cut those apart, put them together into the nucleotides. Remember, RNA is also made of nucleotides. and build your RNA pieces. Then you're going to use one strand of your DNA. So in transcription, the first thing that's going to happen is the RNA or the DNA is going to separate, and the RNA is going to match up nucleotides, but only this time to one strand of the DNA. And remember, U is going to match up to A. And again, you can see where it's going to have those two hydrogen bonds. It's going to match up. G is going to match up to C. And you can tell that these are the RNA because they have the R for the sugar ribose. And then we need a C to match up to our G. So we've made, and you can continue on and make the whole thing. So pause the video and make the whole thing. So now we have our piece of RNA that's going to come out of the DNA. The DNA would go back together and stay in the nucleus. The RNA goes out to the ribosomes in the cytoplasm, and it's going to make the DNA. Now, I don't have a way for you to make a ribosome out of paper, so you're just going to use that chart in your book, which is on page 192, and look up the amino acid sequence that goes with these codons. Remember, these are the codons of our RNA. So they are going to match up with the anticodons of the tRNA, and then that's going to bring the amino acid, which is going to get bonded together to make the protein. So just look up the codons and write down what amino acid sequence would come from that whole DNA sequence that I gave you back in part 1. So I only did part of it here. Look at that whole sequence and write down all the amino acids from that sequence. All right, hopefully that gives you a better feel for these processes that are occurring in the DNA, how DNA gets copied, how DNA gets transcribed and translated into a protein. All right, now do the quiz in Canvas also.